discuss recent understandings related to iron metabolism in dementia. Dementia is an umbrella term that describes a collection of symptoms that are caused by various disorders that affect the brain and its function. The most common form is Alzheimer's disease. Dementia majorly interferes with a person's life. Cognition, emotion, and behavior becomes abnormal, whilst bodily functions are gradually lost. The time between onset of symptoms and diagnosis is prolonged. Early signs of dementia can be subtle, but can progress to frequent memory loss, confusion, personality change, and withdrawal. Dementia is one of the fastest growing conditions in Australia and second leading cause of death. There are an estimated 459,000 Australians living with dementia and expected to increase to 590,000 by 2028. Iron metabolism plays an important role in the human body as a vital component of hemoglobin, the substance in red blood cells that carries oxygen from the lungs and disperses it throughout the body. Iron is responsible for a number of cellular operations such as oxygen transportation, coating neurons with myelin, mitochondrial operation, and neurotransmitter synthesis. APP and iron-regulate proteins regulate iron from homeostasis. Homeostatic mechanisms provide the conditions for optimal cell function by maintaining an equilibrium of available iron concentrations between cellular compartments and buffering molecules, preventing toxic effects. Iron accumulation should be tightly regulated and controlled. Iron can be stored in a non-toxic form or be exported into the circulating blood through transmembrane protein ferroportin. Iron crosses the vascular endothelial cells of the blood-brain barrier, mainly bound to transferrin. Transferrin receptors line the lumen of the brain and facilitate iron uptake into the brain's vascular endothelial cells via endocytosis. Iron is either synthesized in the brain by the choroid plexus and oligodendrocytes to provide support and insulation to axons in the central nervous system, or iron cells continuously move between neurons and astrocytes. The relationship between iron metabolism and dementia remains obscure, despite various in-depth studies. Based on various discoveries related to iron, researchers have realized that iron plays an extremely important role in the occurrence of Alzheimer's disease, and iron dyshomeostasis is increasingly reported as a potential cause of Alzheimer's disease pathophysiology. It is known that excessive iron contributes to the deposition of amyloid and the formation of neurofibrillary tangles, which promote the development of Alzheimer's disease. Abnormal iron homeostasis, more commonly known as imbalance in iron deposition through different mechanisms, can induce cellular damage through hydroxyl radical production, known as the Fenton reaction. This can cause oxidative stress reaction due to its neurotoxic effect, resulting in impaired physiological activity, cognitive function, and behavior as lipids, proteins, and DNA structure becomes damaged. In some cases, this can even lead to cell death. Increased concentrations of iron paired with an aging brain can compromise the iron homeostatic system, leading to an excess of iron that does not officially bond to storage proteins or molecules. The redistribution of iron can be caused by increased blood-brain barrier permeability due to inflammation. The inflammatory state increases in the brains of the elderly, leading to region-specific iron deposition in the microglia, astrocytes of the cortex, cerebellum, hippocampus, basal ganglia, and amygdala. Ferritin concentrations generally increase with age. Goodman's 1953 study found that iron was increased in the senile plaques in those with Alzheimer's. The use of a quantitative susceptibility map once again confirmed the co-localization of brain iron and that brain iron depositions promoted the development of the disease. In 2014, Ward compared two groups of people of the same age, one group normally healthy and the other with Alzheimer's. Iron deposition in patients with Alzheimer's was much more prominent in the substantia nigra, globus pallidus, caudate nucleus, and cortex, as these areas of the brain are closely linked to neurodegenerative diseases. Iron chelators that are able to cross the BBB and penetrate cells have received a great deal of scientific attention and obtained excellent results when tested. Utilizing cloquinol, a study in 2001 by Cherney has found treatment on transgenic mice with Alzheimer's can decrease the deposition of amyloid in the brain and improve memory impairment. Similarly, Holcomb's study in 2008 noted that iron deposits in transgenic mice models with Alzheimer's disease co-localized with beta-amyloid plaques and increases in total brain iron plaque formation. Overexpression of heme oxygenase in mice leads to iron overloading and neurofibrillary tangle formation. Moreover, Stockwell's study in 2010 stated that when intracellular iron affiliates with plaque, it accumulates and activates failure of APP, advancing the onset of Alzheimer's disease. Zhang and Yang's study in 2020 confirmed ferroptosis as the accumulation of redox-active iron and reduction in glutathione level, which plays a critical role in cell death. Interestingly, in 2011, McLaughlin tested deferazimine on 48 Alzheimer's patients to prove a substantial reduction in the rate of brain deterioration. The test compared this with a group of patients with Alzheimer's who were given a placebo. Despite such effective results, no other clinical studies have been reported. Developments in MRIs have been made to detect iron in the brain at an amplified level and help establish variations in iron, even outside the regions of deep grey matter nuclei with high iron concentration. Despite these recent understandings and evidential studies, gaps remain, and information needs to be refined. More comprehensive studies need to be conducted to confirm the relationship between iron metabolism and dementia. For example, testing more iron chelators for potential treatment and establishing more cellular pathways to oversee iron accumulation in the brain with aging.